as the title says, I'm understanding the mechanisms of soil microbial function and their role in cropping systems. And I'm supervised by Terry Rose, Michael Rose, and Lucas Van Sweeten, and all three of those guys are involved with the soil CRC. Sorry, I might just... There we go. That's it. There we go. Okay, so there are quite a few practices within cropping systems in Australia that influence soil biology or microbes in soils. The first is tillage. So conventional tillage, what happens is it's the preparation of the seed bed. It stops the competition of weeds with the microbes. Sorry. And during this time, there's sort of like a burst of microbial activity. What's happened is the soil organic matter that some of those microbes might not have had access to, when the tines have gone in, they've turned up all that soil. The microbes, they just have all this access to all this carbon storage that wasn't in their vicinity at the time, and they just go gangbusters. So during conventional tillage, there's an immediate burst of microbial activity. However, this subsides fairly quickly, and what may have happened is that that tillage might have broken up sort of microbial networks that were pre-existing and working harmoniously and at equilibrium, as we're talking about earlier. And then, so there's conservation tillage, which is as long as about 30% of the soil biomass is left on top of the then that's what's considered conservation tillage. And then there's no tillage, and no-till practices, while are fantastic for microbes because they get to stay in that state and keep working and undisturbed. They also, the carbon pool can become stagnant, and less carbon can be sort of become available, so they can start eating into that carbon pool. Your crop management, so your crop rotation, where you cover cropping, your intercropping and companion cropping. Obviously, monocropping would have a negative effect because it's the same carbon sources going in over the same time. And what that does is that reduces your microbial diversity down to only a small set of microbes that feed off what's being fed by that same plant over and over and over again. And this is how the sort of pathogenic microbes come into play. And at the same time, it's allows sort of diseases to come through, and then precision sowing. So they're starting to realize that if you sort of sow your crops too close together, that same plant is drawing from a common microbial pool, and then the plants start competing with each other for those microbes. And sometimes there's not enough nutrients in the soil to allow the sort of expanding microbiome to service both plants. So they're trying to tap from the same sort of nutrient source. So I know CSIRO and I know DPI are both looking at not only row spacings, but how far apart the plants need to be. And it's obviously different for different crop species. And that's a huge leap forward in understanding that if crops are sown too close together, then those microbes are, feeding off the same plant. And then obviously our chemicals and our inoculants, so herbicides, pesticides, particularly fungicides, can be very detrimental. Now, fungicides can be targeted. However, they can wipe out a large portion of fungi because fungicides aren't necessarily species specific. They tend to be sort of more at the higher levels of a target. So it might be like a family, so you get family, genus, species. So once it hits that one family, that's a whole bunch of different fungi that have been knocked out. Then there's obviously your inoculants, so your microbial sprays. And as we're doing this project, we're trying to figure out whether they actually reach their intended target. But another way to do this is seed coating. So seeds are coated with a microbial inoculant, and then they're there from the start, from germination. Yep.
Cameron,
sorry.
can I just ask a question about if you go back to the other slide with the chemicals and inoculants?
Yep.
We're getting a bit of a shift toward because of the concerns around microbial activity in the soil and understanding their function in cycling nutrients and making some nutrients available. We are getting a bit of a shift towards some landholders not wanting to use fungicides like seed dressings in particular. So and then also this so that's sort of one question, I suppose, if you'd like to consider. And also these microbial sprays and seed coatings. So I've got landholders who are, mixing up their own inoculants or extracting, worm extract out of composts and using that as what they're calling as an inoculant or a seed coating. So we're getting this language being used for different, traditionally seed coatings were fungicides or rhizobium inoculant, and now people are mixing up their own, what they're calling seed coatings and seed dressings, which they're believing is adding biology and
to the seed,
to the soil for a benefit of nutrient cycling or boosting the microbiology in the soil. I mean, do you have a comment about that?
Yes, that's I guess seed coating for me isn't what's being used, it's the process. So you can coat your seeds in anything you like, and to say that your seed coating doesn't have any connotations, both positive or negative. It's simply you are coating your seeds with something. Now, I know that it's particularly in the wheat industry, it's very common to coat your seeds in something like gaucho, just to protect the seed for those early germination times. And that does have an effect on the microbiome immediately around the seed. But you're right, if you were to coat it with a sort of microbial community, and you're talking about they're not using fungicides anymore, or they're coming up with their homemade microbial inoculants. As you're about to find out in a couple of slides later, unless they specifically know the diversity profile that they got to in their soils to begin with, you don't know that the microbes that you're adding into that soil benefit that soil type and have the right conditions for those microbes that you're adding. So you might add those microbes in, and then a week later they're all gone. They didn't survive. It's the wrong type of conditions for the microbes that you've added. They might go gangbusters. You don't know until you've taken a sort of a sample and you've tested that microbial diversity to start with, and you're sure that the microbes that you're adding are sort of able to withstand the conditions or the crop management practices that you have, as well as the soil type. Then it may last that one season, and then if you don't put a cover crop on, that particular microbe that you've added might not survive because it needs a constant feed of nitrogen or carbon, or it needs it might be an AMF fungi, so it needs a root system. And if it doesn't have a root system to hang on to, it's gone within the next growing season. And then you've sort of run into this same problem with farmers of having to put chemicals on chemicals. If you don't look after your management practices, you might have to keep adding your microbes in your soil. And as we're starting to find out at the moment, that microbial inoculants aren't cheap. And I think it's on average, about $40 a litre. And so you still got a cost there to add to your crop. I think, so it's the Australian Inoculant Research Group at EMAI. They've done a approximate calculation that if we were to stop using nitrogen fertilizer and switch to complete biological fixation of N in our soils, it would save Australian growers to the tune of $4 billion. So it's great in theory to make that switch, but whether that happens in real time, that's easier said than done. It's the analogy I use is like trying to convince Australia as a country to switch to solar energy. And we're dealing with something much more complex than a solar panel. We're dealing with a live, forever changing dynamic system of microbes. So to convince that the whole of growers to no longer put, sort of fertilizer into their soil and make it completely microbial, that's a huge task. And at the same time, we're still not 100% sure that it's going to work based on because each growing site is different, each management practice is different, cropping types are different. So there's almost has to be a separate microbial plan per grower, per site. So even a field that's about 1 kilometre away would have a completely different microbiome, different soil types, different, and that's going to affect what microbes you should be putting in your soil. And so the soil microbes are involved in these are the sort of key soil functions. So you got the biochemical regulation of plant hormones and diseases. You got your soil organic matter, turning over of organic matter and carbon storage. Nutrient cycling is probably the biggest one. Soil structure. So they're involved in sort of aggregation, and then soil water availability. So I was just going to go a little bit into not exact mechanisms of how they do this, but just a brief overview. So if I lose you here at any point, just stop me and I could sort of go, further explain it. So not all microbes are beneficial. And if you have a sudden or increase in microbes in your microbiome, in your soil, the chances are that there's going to be pathogenic bacteria and fungi. Same with the monocropping. So if you sort of crop over and over and over and over again, eventually there'll be pathogenic fungi and bacteria that go, hang on a minute, I really like this food source, and they start, out competing. But there are obviously beneficial ones. So you've got your plant growth promoting rhizobacteria, and as you can see here, they come in sort of the Pseudomonas, Bacillus, and Serratia varieties, and they prevent pathogenic bacteria through nutrient competition. So PGPR are very good at outcompeting pathogenic bacteria. They tend to be more efficient in taking in soil nutrients. Then you got your AMF. Now they tend to attach themselves to roots, and they stimulate a phytohormone response in the soils. Now these phytohormones then develop what's called systemic resistance, and the bacteria and the fungi can no longer sort of take in those nutrients, because the plant, again, the mechanism is still a bit understudied here, but the plant can recognize that these are bad bacteria, bad fungi, and I'm not going to feed those. I'm only going to feed the ones that are helping me. So it's actually quite smart how the plants do this. But the exact sort of mechanisms and genetic releases are still unknown. And then you've got actinomycetes. So actinomycetes are halfway between bacteria and fungi. They're kind of like a middle ground. And in both structure and function, they kind of do a bit of both. Now, actinomycetes are known for what's called pathogenic antagonism. Now, they'll try any way they can to stop the growth, the reproduction, or even the function of pathogenic microbes. And this can be done by, again, all the previous listed ways. So systemic resistance, nutrient competition, parasitism, so they can actually eat the fungi or the bacteria themselves. But the most common way is antibiotic production. So they take in the nutrients, and then as a byproduct, they produce these antibiotic compounds, which then attack the pathogenic bacteria. That being said, it's not always specific to just the pathogenic bacteria. So sometimes this antibiotic production can affect the positive microbes as well. So then we're moving on to sort of soil organic matter and carbon storage. So microbes, their first and foremost role is decomposition. So you've got your bacteria decomposers, and they tend to be the first ones to have a crack at your soil organic matter. So the most common that I found in literature is Bacillus subtilis. It basically has an enormous array of functions, but one of its big time players in decomposition of soil organic matter. Fungal decomposers don't necessarily directly always try and decompose the organic matter to begin with. They like to feed off sort of root exudates. So it's different carbon forms that are secreted or exuded by the roots. And then they utilize this to produce enzymes. And these enzymes then act on the soil organic matter and it further breaks it down. At the end of all this, you can still have some complex soil organic matter left in the soil. And this is where actinomycetes come in, and they can basically further decompose the organic matter, or if it's a complex carbon form, they'll act on that. This ends up with a soil organic carbon pool in the soil, and plants can take this up, and other microbes utilize this for their functions to go on and do other things. Now, there's some debate amongst sort of soil carbon experts about the manner in which this soil carbon exists. So you might have heard humic substances, and they're starting to debate whether those substances are actually a final resting place. What they think is it's more likely that the type of carbon that ends up in the soil is forever changing. It's fluid. It doesn't sort of finish in a final spot and goes, that's it, I'm just going to stay right here. As you can see, that the soil carbon is used by other bacteria, and there's a lot of different processes and different forms of carbon for it to end up as organic carbon just sitting in the soil. Some of that carbon is lost in the soil from leaching, so it doesn't always stay. Just because you started with this amount of carbon doesn't mean you're going to end up with that amount of carbon. All of these processes, microbes go through what's called microbial respiration. So they respire like us, and they produce carbon dioxide. So this then gets released into the atmosphere. Again, the whole, you've probably heard a lot about carbon sequestration and how good it is. If you suddenly increase the amount of carbon sitting in your soil rapidly, the microbes go, especially the R stratus, which I'll talk about a little bit later, go, thank you very much. I'll take all that carbon, and they rapidly sort of reproduce and expand. And then you get this influx of actually CO2 in the atmosphere. So that's because the process of them breaking down that carbon, they actually produce carbon dioxide as a byproduct. However, for the plants, this is really good because then they take in that carbon dioxide and they release it as root exudates, and we start the cycle all over again. So this is known as carbon turnover. So it's sort of the forever moving parts of carbon in soil. So in terms of nutrient cycling, carbon is obviously one of them, but another big player is nitrogen. So in your soil organic matter, after it's been decomposed by bacteria and fungi, you end up with organic nitrogen. And this is generally in the form of sort of genetic materials, nucleic acids. This is then mineralized by bacteria into ammonium. And however, other microbes utilize ammonium for their processes. And so therefore it's then immobilized back to organic nitrogen. Now, the reason why they use immobilization is because plants don't take up organic nitrogen. They take it up in the form of ammonium. So it's their most common usage of nitrogen for uptake. Ammonium can then be go through a process called nitrification, and this is done by a particular set of bacteria called Nitrosomonas. And then that you end up with nitrite. Now, nitrite, like some of the carbon in a carbon pool, can be lost through leaching. But further nitrification, so if it's gone through one set of nitrification, another bacteria called Nitrobacter then acts upon that and further nitrifies, and we end up with nitrate. This nitrate can then be assimilated into the roots. So although plants prefer to take it up as ammonium, they also do take up nitrogen through nitrate. At the same time, proteobacteria can then act on that nitrate, and it's a pretty complex process, but it's called dissimilatory nitrate reduction to ammonium. So this nitrate is then converted before it gets taken up by the plants back to ammonium, and then so it's just like an added steps for the plants to access this nitrogen. Then there's denitrification. So Pseudomonas denitrificans. They've really scraped the bottom of the barrel for the naming of that one there. Basically, we end up with atmospheric nitrogen. And this is not the end of the nitrogen cycle. So this atmospheric nitrogen can then be fixated directly into the roots by rhizobium. And that's probably your most common. So your legumes have these sort of bacterial nodules on them, the rhizobium nodules, and they fixate the nitrogen directly into the roots. But there's other ways as well. So there's a free living fixation, which it's called Azotobacter. Azotobacter, rather than fixating it directly into the roots, converts the atmospheric nitrogen and turns it into this ammonium in the bulk soil, and then the roots take it up. So it's just an added step. And as you can see, that the microbes are doing everything they can in some way to get the nitrogen into a form that the plants can take it up, or turn it into atmospheric nitrogen, and then the plants will either directly fix that, or it'll end up back in the soil or back into some other organic matter, which will then go back into decomposition and back into sort of organic nitrogen. Now, soil structure is a big one. It wasn't until the last probably a couple of decades that they've realized that microbes are so involved in the structure of our soil. So a lot of it was, credit was given to plant roots. And yes, plant roots do a large amount of stabilization of soil structure, but soil aggregation, a lot of that work is done by microbes. So plant roots exude different forms of carbon in root exudates, and obviously through the breakdown of soil organic matter, you've got different soil nutrients sitting in there, and these are acted upon by various microbes, both bacteria and fungi, and then those are broken down through the decomposition. They're broken down into secondary metabolites, and these are then used as binding agents or coagulants, and what you end up with is your soil aggregation or an aggregate. And these form sort of little pockets of clumped nutrients, but also water, basically anything a microbe needs to sort of live and survive. And same with the roots. The roots like to tap into these aggregates. Another way that soil structure is affected by microbes is through fungal enmeshment. So you've heard of hyphal networks, but fungi don't necessarily have to form these long linear lines. Sometimes they form like a mesh, like a gauze mat. And basically what they're trying to do is they're trying to increase the surface area to volume ratio of the plant root. So they'll tap into that plant root and they'll spread themselves out as thick and as wide as they can. And basically that allows, if there's a nutrient-rich pocket in the soil, they'll just completely surround that, and that plant will have constant access to that nutrient. And the fungi actually operate as fantastic gatekeepers, and they sort of take in the nutrient as much as the plant needs. They won't take any more than that. They don't take any more than they need either. So these enmeshments are fantastic for plants. However, as we were speaking earlier about tillage, these enmeshments can be just sliced in half through tines going through the soil. So sometimes it's good to break up the soil and give access to this carbon storage, but sometimes these hyphal networks have taken time to develop over time, and then we've just cut them in half. So, sometimes it's a win-lose situation, and it's up to the specific grower, and depending on what kind of soil they're dealing with, whether it benefits them to turn that soil over or not. The last sort of key function for microbes is soil water availability. Now, that aggregation that we're talking about obviously allows these pockets of air in the soil, but it also increases the ability for water to move through the soil. So if you have a hard top of soil that hasn't been affected by microbes, that water's not going anywhere. Sometimes the plant roots can't sort of access it. Whereas if you have a nice porous soil water moves fairly freely through the soil. Now, it's not only plants that need water to function. Microbes are more often than not hydrophilic, so they like water. You do have a few hydrophobic microbes, but again, they're more extremophiles, so they're not necessary unless the environment sort of promotes their growth. And there's a varying amount of sort of water availability that microbes benefit from. So again, like your carbon inputs, your nitrogen inputs, all that sort of stuff. If you have a climate that is, high rainfall, then the microbes that are more hydrophilic, so the ones that love the water more often, are going to thrive. If you have a more sort of a drier region or climate, then the ones that don't like a lot of water and are more resilient to utilization of less water, then they're the microbes that you're going to benefit. And this goes back to me saying about how people will just put any microbe in any soil and hope for the best. You actually need to specifically target what your climatic conditions are for the soil, because your microbes may not survive what you're adding in. So a lot of talk about fungi and bacteria, mainly because they're the two major players in the soil, but there are other microbes at play. And so your protozoa are very important they feed on bacteria and fungi. So they're sort of not just the pathogenic bacteria, but they do feed on sort of the beneficial ones as well. But it is important for protozoa to exist so that bacterial predation, the feeding on the pathogenic bacteria and fungi to stop their growth. And then by feeding on these, they allow for a different form of nutrient to be available to other microbes. So they eat the bacteria and then convert the cells into sort of bioavailable nutrients. Algae are very important. They're seen as the pioneers of the soil world. Algae are autotrophic. They are themselves a source of carbon and nitrogen for other microbes. So they sort of attach themselves to anything they can, and the algae will grow, and then the bigger microbes will then feed on the algae. And then nematode, again, nematodes are very similar to protozoa in the way they act. They're very important for sort of the regulation of bacteria and fungal populations in the soil. But also, because of their size, they do affect soil aggregation. So they create these pockets by sort of moving around in the soil and pushing the soil around so that they can expand themselves. So spatial influence of soil microbial diversity. So again, depending on where you put your crops in and how big a root system your crop has, and also how close your crops are together, will influence the microbial diversity that you have. Within just a single plant in the soil, you have the bulk soil, which is on the outside, and that will have, again, one certain type of microbes that only function with nutrients in the bulk soil. Closer to the root is the rhizosphere. Now, this has been known. So Lorenzo Hiltner was talking about the rhizosphere in 1904. So the idea that microbes have been involved in plant growth is over 100 years old. Now, new light has come to what's called the rhizoplane. So this is basically the immediate sort of soil particles or nutrient particles that are semi-attached to the roots but aren't necessarily penetrating the roots. They're basically the sort of like the gatekeepers of nutrient release to and from the root itself and the rhizosphere. And last, the endosphere. So the endosphere are more like your arbuscular mycorrhizal fungi that sort of attach themselves not only just around the root but into the root itself. So they actually penetrate the cells of the root to achieve sort of nutrient exchange. Temporal influence of soil microbial diversity. So again, this comes back to soil microbiologists trying to marry up terms with sort of terrestrial ecologists. So there were, before sort of looking at it as a time point of view, it basically was copiotrophs, which is microbes that benefit from large amounts of nutrients to microbes that can function with like low amounts of nutrient availability. So previous to looking at it as like an R strategist or a K strategist, it just used to be about nutrient availability. Nowadays, they're trying to utilize the same terminology as you would with terrestrial animals or plant life. So our R strategists are the ones that grow very quickly, very early on. They need lots of nutrients, but they're not very good at sort of fighting for those nutrients. They tend to be in large numbers, and they don't require a lot of energy to reproduce, to do their function, because they will just overpopulate themselves. So their expenditure is low. K strategists, again, are your longer developing microbes. They take a while. They're very good at competing because they will utilize the smallest amounts of nutrients to reproduce and to do their function. There's not as many of them because they're so efficient at their job. But however, they do tend to take up more nutrient in terms of for their sustained life. So because they live for longer amounts of time, they tend to be more expensive in terms of, to keep them going, you need to put in more nutrients. So new technologies have allowed us to sort of shift into different ideals of how we approach microbial research. So we have culture dependent versus independent. Now, only up until the late 90s, people were still taking soil samples, taking a bit of and streak plating, growing the microbe out on an agar plate, and then testing that microbe. Nowadays, we can literally just take a soil sample, we can look at the nucleic acids and go, this is the microbe that is in that soil. So that's a huge jump in the way that we look at what microbes are there. This genome sequencing technology is forever improving and becoming more accurate. The computational power, however, to match that and to figure out and do the calculations of what's there and what's not, and to say with certainty whether there's ever any overlap in the genetic material, is lacking. And so we're at a stage now where we have sort of gene sequencing technology that needs a supercomputer to be able to run it and say with absolute certainty that that's what's there and that's what isn't. There's obviously human error involved as well in these samplings, but the biggest limit to in terms of microbial diversity is the computing power to run the statistical analysis of those samples. Before we were talking about sort of the function of soils. So there's bioassays now that exists where you can literally take a section of soil and quickly run it on a microplate, or there's a box called MicroResp, and that basically will tell you if the microbes in that soil are respiring. So if they're doing their job, if they're functioning, as I showed you earlier, that they're producing that CO2. MicroResp can calculate how much CO2 is being produced. So if you know how many microbes are there, or how much or what type are there, and you measure that CO2 production, you can actually figure out how efficient those microbes are at doing their function. It's hard to tell sometimes what exact function that is, but at least you know that they are alive, they are doing some kind of job, and by how much you can tell by the amount of CO2 that's produced. This is just a little bit of a snapshot to give you an idea, and I don't want to scare you too much, but this is like a needle in a stack of needles. So this is a meta-analysis paper that I had a look at, and these guys just looked at one lot of bacteria, three different phylum, which is like pretty high up in terms of the bacterial domain, but they only looked at three. And then within that three, they only looked at a few classes. And then within those classes, these are the amount of species that were involved in different crop types. So as you can see here, your Bacillus subtilis is found in three different major crop types. And this is all Northern Hemisphere, but a lot of the times the bacterial species here in the Southern Hemisphere if we're growing those same species types, we're going to tend to find the same bacteria on those plants. And as you can see, bacillus is found in almost every type of major crop that can be grown. But at the same time, you can see variability amongst the type of bacteria that's grown. So sometimes specific bacteria will favor one crop over another. But in that same breath, you can get one crop type that will have, a huge array of different bacteria, all doing either the same function or a completely different function. So that brings me on to my next slide, is that the way we used to approach microbial research, and growers used to have this same mindset, was if you were to increase a population of one species of microbe, that you increase its function and then you increase the soil function. So the microbial function might be a specific step in the nutrient cycling process. The overall soil function is nutrient cycling of itself, or plant uptake of that particular nutrient. Now, if you increase one microbe population, there's no way of telling that it's directly involved. Like if you're unsure what microbe you're adding, or if the system will allow that microbe to do that. So sometimes both researchers and growers have gone, I just sprayed my crop with this microbe inoculant, now look how much nitrogen I've got in my soil. You can't always say that specific microbe was doing that exact role, that the increase in nitrogen can be attributed to that one microbe. So it's actually a lot more like this. If you increase your diversity, if you increase the different types of microbes that you have right across the board, then you're going to increase the microbial functions. Now, as you can see here, multiple populations or species of microbes can be involved in one function. That same function one species can be involved in two separate functions. And then the overall soil function, which might be, again, going back to those key functions, so you get your nutrient cycling, your carbon storage, your biochemical regulation, that can be affected by a whole suite of different microbes all interconnecting. The last part is what we're starting to realize now is that microbes can actually pass on genetic information sideways. So they can pass it on from one species to another without having to reproduce. And this is huge in the microbial space, because if one microbe has learned to do something and another microbe is also involved in that same process but are lagging behind, but they aren't too dissimilar in terms of their genetic makeup, but they are different species, that microbe can pass on that genetic information to a different species and teach it how to function better, or how to better adapt. Or it may be a regulatory process in saying, oh, hang on, we've done our job, there's no more nutrients left, and it will pass that genetic information going, we need to stop, we can't grow anymore because there's too much. So that changes the way that we look at what microbes that we need to put in our soil, because it's a much more complex picture than first thought. And so what the new approach to soil microbes is increasing its diversity will increase its ability to function in the soil and become more resilient to outside or external pressures. Just a quick overview of my experiment that I'm looking at is I'm going to inoculate some soils, and then I'm going to reintroduce some microbial communities to those to the sterilized soils. These sterilized soils will have no microbial life left. They won't be doing any activity because of the gamma radiation. Once I re-inoculate them, I'm going to see whether, and again, I'll have different soil types, and I want to see whether the soil type affects the microbial community over sort of 0, 7, 28, and 90 days. So at 28 days, I'm going to send off my soil samples to AGRF to see whether they're going to send me back the genetic information. And I'm going to see whether that microbial diversity profile has changed based on the soil that it was inoculated into. But all along that time period, I'm also going to be testing the functions of those soils and the physicochemical properties of the soil to see whether not only the soil type affects the microbial growth, but whether the microbes can actually drive and change their environment they live in. Because as you've seen before in the slides, quite a number of soil nutrients, the physical structure of the soil, water availability are all driven by microbes. So it's suggested that there's a feedback loop involved with microbes and the soil, and they eventually, given enough time, will reach some kind of equilibrium. I'd just like to thank sort of Soil CRC, Southern Cross Plant Science, the Centre for Organics Research, and the DPI for allowing me to do this research. Thanks, guys.
Thanks, Cameron. That was really great. And Misty's joined us. So welcome, Misty. Thanks for logging in. Nice to see your face. I've just got a quick question about that last slide. Can you just tell us what AGRF is again? I don't know if you...
Oh, sorry, that's the Australian Genomics Research Facility. Okay. So AGRF, basically our country's version of if you're not going to do your own DNA testing. I mean, Saudi have started to do their own stuff, but quite often Saudi and AGRF sort of utilize the same structures and equipment. They even sometimes pass on work to each other. So they kind of operate together. But yeah, AGRF is sort of the overseeing body of genetic research in this country.
Yeah, great. And what sort of physiochemical testing will you be doing?
So I'll be looking at mainly, here at Wollongbar DPI, we have a soil testing unit, and there's a package that you can test sort of so your carbon, your nitrogen, your phosphorus levels. I'm going to look at soil pH, soil temperature, because with microbial activity, so respiration, like most living organisms, if you are respiring or doing some kind of activity, you tend to give off a bit of heat. So a large abundance of microbes in the soil would slightly raise that temperature. Again, I don't know how accurate I'll be able to get it, but it's still some kind of indication of microbial activity. But the big ones I'm looking for are obviously sort of the NPK sort of levels, you know, carbon storage, pH. Again, pH, the slightest change in pH levels in soil can completely alter the microbiome. So microbes are more often than not very sensitive to pH. So, and you can, and I'm talking within one sort of degree of a pH. So if you go from 6 to 5, you are completely changing your microbiome. Even 6 to 5.5 is a big enough change to affect quite a number of microbes in the soil. Now, that doesn't necessarily mean that you're going to completely wipe out those microbes. You might be just reducing their function. At the same time, by reducing your pH or making it more alkaline, you could be bringing in a whole new set of microbes that benefit from that change in the pH. So that's why I keep banging on about this diversity profile. If you increase that diversity, you're increasing the ability for the microbiome to function depending on the external pressure that's on those soils. So the more different types of microbes there are, the better guarded you are. So it's like how we get vaccines, right? If we get just the one vaccine, we're not protected from all the diseases. If we get a heap of vaccines over and over and over again and different types of vaccines, our body is better equipped to defend ourselves from a large array of diseases. Microbes are the same in the soil. The more different types of microbes they are, the increase the function you get, the better resilience you get. And again, it doesn't always translate to a beneficial sort of yield result. What happens more often than not is a less fluctuating yield. So rather than a big roller coaster of yield, it starts to flatten out. And again, I'm not saying that it completely flattens out, but you might have lower highs, but you'll also have higher lows, and it'll sort of bring those dips back into play a bit. And that's that's the benefit of looking after our soil biology. Is not necessarily an immediate benefit to your cash crop, more of a long-term benefit to your whole paddock, not just the one crop.
Do you think fine if you don't know, because I don't know. Do you think that the soil carbon process with microbes, chewing up carbon and nutrient cycling and those sort of things, do we know what sort of effect that's having on pH? I mean, we know that if we add certain types of fertilizers, that they can be acidifying. That process that you showed with the carbon cycle and going through the different nitrogen processes, is that in itself, I mean, is there some sort of acidifying aspects to that, just in itself, that just those chemical processes?
So again, it may not be acidifying. It could be making the soil more basic or alkaline. So certain species of microbes have varying functions. So as I was showing before, one microbe might have multiple functions, or multiple microbes might all perform the same function. Those functions, you're right, can produce acids. They can produce complex carbohydrates. They can end up with certain salts. It might not be the salt that not your table salt, but a salt, and those all have varying effects on pH. And again, if you increase your diversity, you might have one group of microbes that are making it more acidic, and then you'll have a different group of microbes that are making the soil more alkaline, and then they'll balance out. And so, again, it's not necessarily about if you pump in a specific set of microbes into that system, into the soil, you might be throwing that completely out of balance, because you might be putting in a microbe that loves acidic environments, and you've just given it a whole bunch of carbon. It's like, here's the carbon, here's your fertilizer, here's your soil, go for it. And then you might produce a more acidic soil, but if you've got a crop that benefits from a slightly more, I'm not saying acid soils, I'm just saying a slightly more acidic soil, if that crop benefits from those acid soils, that particular microbe that you just added, you're probably going to see a yield benefit, or just some kind of biomass benefit. And then the grower then thinks, right, I've added this specific microbe and I've got my extra biomass. That's exactly what I need it for. That means that microbe does that job. What the grower doesn't realize is that microbe then might be outcompeting a whole array or whole suite of other beneficial microbes to do that one job. You've just completely thrown out the balance. So while, yes, there is an immediate benefit to that crop, there could be a long-term detrimental effect where they then need to keep putting the same microbe over and over again, because when you introduce a microbe, sort of like you spike it or you inoculate it, that microbe doesn't necessarily know at that stage how to reproduce itself based on what's available to it. And so it might completely deplete by the end of the growing season. It might deplete within a couple of weeks. You don't know. And so we've got to stop thinking of microbes as like the same way we look at chemicals, in that we can just spray the soil and these microbes will then stay in there and do their job. Terry, one of my supervisors, he's a project leader for plant-based solutions. And the idea is that all these different cover cropping, intercropping, rotational cropping, these processes naturally diversify your microbes in your soil. And farmers are starting to catch on to this now. Growers are starting to realize that if I sort of grow a legume after my my canola, I'm gonna fix some nitrogen in there, and I don't have to. Again, they're not completely wiping out the need for nitrogen, but they are drastically reducing how much nitrogen they need to put on the next time they grow some canola or wheat or whatever. So the same thing's happening with the intercropping, is that growers are realizing that sometimes if I don't have a rotational option, sometimes if you grow two crops at once, either in the interrows or in between the crops themselves, and they just don't grow as high, you can still take off the wheat head or the canola or whatever, but you've got another crop sitting underneath that is adding microbial diversity to the soil. It's not taking too much nutrients out. So there's quite a few cut like grasses or even other types of crops that don't require as much nutrients to grow, and they're very resilient. And therefore, and farmers are using various ways of dealing with those intercrops, where they spray them out or they sort of mow them in, and then you're just increasing that carbon into that soil. Again, that might have an immediate effect, but you don't know what it's doing. We still don't understand what it's doing long term. So more research is needed into these. Again, we still know that they're beneficial for now. We just don't know how beneficial or for how long. So we're still trying to find out whether these crop management practices have a huge effect. So we're starting to realize that you can increase your microbial diversity, even if it's only by sort of 2.5%. That 2.5% could be gangbusters in terms of results. Also, if you're increasing the abundance, you might increase the abundance of a microbe by 40%. That's huge, but it might not do anything. The microbe just might feed on the same nutrients and then just become stagnant or dormant. So, yeah, it's an interesting way to look at it.
Mm. Yep. Yeah, great. Do you guys have any questions? Because I could just keep asking questions all day.
Yeah, I got one, Cameron.
Yep.
So probably whether we talk about it now or we can talk later, just give some thought to next week. So interesting observation for the mung beans, which we're going to look at next week. So where we had applied the endofuse seed treatment as a liquid, as endofuse as a liquid in furrow treatment. We noticed within the previous crop, such as whether it was either the wheat, the canola, or the fallow, so within that factor, where we had applied the endofuse, the crop performance was significantly lower than anything else. So I'm interested to your ideas, what potentially could be happening. So to me, it looks like whether the endofuse has somehow looked out, let's say the rhizobia, so end fixing.
Yep.
And it's just like absolute chalk and cheese.
So
spoke to Sumitomo about it. So just give some thought to what could be going on, but also whether or not we need to think about the way we sample those mung beans next week. So whether or not you guys have the capacity to say assess the nodulation, end fixing, I don't know, but there's something definitely going on.
Right. So Felicity was just asking what endofuse actually is. So, sorry, do you want to?
Oh,
you guys.
Sorry.
Yeah.
So endofuse is a commercial product manufactured by Sumitomo, which they're recommending, I think, on a range of crops. I'm just not sure of the label off the top of my head, but it's, in my mind, and you can correct me, Cameron, it's a mixture of basically AMF species, of which you can apply either to seed or as a liquid in furrow treatment to crops. And that's certainly been a treatment which we've included in this trial we've got down at Lally.
So AMF is your arbuscular mycorrhizal fungi, and as I was showing you a bit earlier, they exist sort of both in the bulk soil, but also they are the kings of the endosphere. So in terms of their ability to penetrate root systems and create these sort of hyphal networks and increase nutrient uptake or root exudate sort of movement. At the same time, the soil structure revolves a lot around AMF. So what Brendan was asking is that in the sites that they've added this AMF inoculant, that they've found reduced growth in that like physical growth, I'm guessing, of the root itself, but also of the nodules, So what's happening is essentially it's a little bit of nutrient competition. So fungi and bacteria can vary with some degree of what nutrients they take up, but largely there's a lot of overlap. And so again, with the fungi, some have sometimes, in terms of their ability to penetrate the root system, they'll fixate onto the root physically, and they can then act as like a barrier between the root and the rhizobium, who are trying to also attach themselves to those roots as a nodule. So think of it like, I don't know if you go back to sort of your high school biology days where you have substrates and substrate inhibitors. So it takes up that binding site. Think of it that way in terms of, but on actually a little bit of a larger scale. So that binding site to the root, the fungi can actually attach themselves to the endosphere of the root, and then the bacteria is trying to do the same, but it just can't. It can't attach itself. But also, if they get there first, and which is the case especially if you do it as a seed coat, I mean that you've already put the fungi on top of the seed, so it's always going to be its first sort of buddy, if you will, in the soil. It's going to take all the nutrients around that, and it'll feed some into the root, because fungi aren't necessarily always parasitic, so they love to sort of benefit their host because it allows them to survive. So they'll go, here's some for me, here's some for the root, here's some for me, here's some for the root, and they'll do that, and they'll hold on to it very tightly. Whereas the bacteria that are sort of in that bulk soil that are somehow trying to get to this new root system, they've got nothing to feed off, so then their numbers reduce. So the population of the rhizobium will reduce, and then they don't have the numbers. Think of like a big standing army. They don't have the numbers to try and take on the fungi that have the AMF that are attached to the roots. So it's not any one process. Same thing could also be sort of systemic resistance. So that fungi could be telling the root, hey, I'm the only one you should be feeding because I'm looking after you. Do not allow anyone else to sort of take any nutrients from your roots. And so it could actually be a phytohormone response in the roots, that even if the rhizobium do find a root to attach to, the root's not going to feed it. So plants have like a way, like you got an arm, you got like a leech on your arm, and you don't want it there. They don't cut off the arm. The brain could just switch that arm off, and there's no blood flow. There's nothing going in. There's no nerve centers. Your arm will just literally die of necrosis. And so the leech is trying to suck out your blood, but there's no blood flow into your arm. You've actually just sprouted a new arm underneath. And so that's just, roots operate in that way at a much more efficient rate than anything that we can kind of conceive. So in terms of the project, Anders and I, like, again, don't select the specific roots that you think have, done worse or better. We still want it randomized so that we can say, well, out of a plot, we picked out, 3 different plants in that plot. This is what we found. And more often than not, there's less nodulation from this endofuse. And again, you're starting to notice this on a sort of a legume type crop where your rhizobium tend to flourish. We don't know if this is happening with non-rhizobium bacteria. So endofuse could actually be, and this is why I say I'd love to see what's happening in the wider community. There could be other bacterial colonies that are at some kind of detrimental effect because the fungi take it over. You've just increased the fungi community tenfold with this endofuse, and the bacteria going, well, hang on, what about us? And so it might not just be rhizobium that are folding. It could be other plant growth promoting bacteria that aren't getting what they need. So that's what I'm more interested in. And so this mung bean crop sampling, I'll be watching it very closely because I'm very interested to see how it might not necessarily be stopping the rhizobium from attaching completely, but you could be reducing the size of those nodules. Even reducing the size of a rhizobium nodule decreases its ability to function as a nitrogen fixating nodule. So, because a lot of the underlying formula for all biological samples is surface area to volume ratio. You increase your surface area to volume ratio, you increase your ability to do whatever function is in a biological system. So the bigger the nodule, the more nitrogen it can fixate. So yeah, I'm very interested to see what we get out of this.
All right. No, I'll just give us some thought then whether we need to do anything different, as in do we need to take more plants or sample some way differently?
Probably take more plants in terms of maybe increase it to say 5 plants per section. And worst case scenario, because Anders and I are keen to do some root scanning. We'd love to see whether these things are having an effect on root morphology. So we've just built a machine here at DPI, and we just lay the root down and we scan it, and then we're going to chop it up into different things, and we're going to have a look to see whether the AMF are penetrating different sections of that root, and then we'll have the overall scan, and we can see whether sort of physically, spatially in the soil, what sections of the root are the most susceptible to the AMF penetrating. So yeah, definitely probably grab a few more. I'll get Anders to double check with you. I'll probably pop down and see him after this, and I'll get him to send you an email. But I reckon if we take a few more samples, we're not going to be selective with the roots that we send off for testing, because we don't want to be biased towards our results. So we just take random roots, and then that's what it is what it is. Whether it's a positive result, negative result, it doesn't matter. But to have extra roots to have a look at, and we can then do other testing on it, that's where I come in. I'd love to see what effect it has on the rhizobium nodulation, root morphology. That would be very interesting. So it probably won't be too many more plant samples than we need. Are they big root systems?
No, mung beans won't be.
Yeah, that's good. So if they don't take up a lot of room, then we can probably just grab a few, because we won't have a lot of room in the car, because we couldn't take all the samples back last time.
Yeah, you'll have to bring a trailer. Make sure you bring a trailer.
Oh, I think that's what we're going to be doing. So that'll be a fun drive over the Ebor Ranges with a trailer hanging behind us.
Very quick one.
Yep.
In the name of time. So other projects, which obviously growers are more than interested in looking at the moment, is the, let's say, the impact of deep ripping. Okay. So this is probably on the back of the last 3 years of drought. So initially people were talking about, I guess, deep ripping, let's say, to try to increase surface roughness. To obviously try and improve our fallow efficiency, so we can obviously store more water and grow more crop. But on the back of that, I guess, so should I say, so we've got some large scale trials which have been set up by growers with their own commercial equipment, where they've gone through and practiced deep ripping. And this is generally done after, let's say, a chickpea crop. So they've got very minimal ground cover to begin the fallow anyway. So they've really got nothing to lose effectively in terms of ground cover. It's generally on those soils which are, I guess, those harder setting red soils, as opposed to those lovely black cracking clays that we tend to work on out here. So that's all well and good to go and do that. But I guess from our point of view, so from a research point of view, it's very hard for us to pull apart or tease apart, I guess, the factors that could be coming into play. So in our mind, it could simply be, is it compaction? Is it a plough pan? And we've just physically gone through and broken that up. Is it, are we causing a release of nutrients by physically aerating the soil and, causing a little bit of nitrification? We've spoke about fallow efficiency. So is it just basically roughening the surface so that we can obviously capture more rain and get into the soil? Or potentially, is it an impact of soil biology by, once again, aerating the soil and disturbing the biology and, like you say, creating a shift in whatever way to some form of biology? So I guess at the end of the day, if we go and do all these trials, which will obviously run over at least 3 or 4 or 5 years, and we can then stand up and say, yes, by deep ripping, over the last 3 crops you've improved your yield, but we still can't categorically say what actually it's being caused by, because it could be combination of all those 4 factors. Any ideas in how you can try and pull apart or pull out the, let's say, the soil biology aspect of it?
Well, so you kind of answered your own question. It's actually a little bit of column A, B, C, and D. So soil biology is extremely sensitive to physical structuring. So if you physically move your soil around, that's going to affect your soil biology, because that's going to allow carbon deposits that weren't accessible by the microbes prior. So if you're especially dealing with a solid soil that does, like a hard top that does not move, microbes can't necessarily penetrate tough soils as any better than, say, I mean, they have a more efficient way to move than roots, but their way of moving through soil is to grow and die, and then grow and die, and then grow and die. They don't necessarily swim through the soil like you think they do.
Yep.
They can attach themselves to water molecules and move that way. But to give you an idea, 1 gram of soil has about 100 million to about 1 billion microbial cells. So that's just from 1 gram of soil. So the way that they move is literally growing a bit further, and then the bit they're trying to move away from, they'll just die, and then it'll just go that way. By deep ripping, so I'm guessing deep ripping, you're going beyond 40 centimeters.
Yeah, so these guys are using just a straight shank and tine, and they're going down to upwards of 400 mil.
Yeah, and so I think sort of spatial, again, sort of you do soil layering. So if you have a look at the different horizons of the soil, you'll get different soil biology. So the top soil, that first 5 centimeters. So Jason Condon, I think, at CSU has done a bit of work with this as well. But he's looked at soil chemistry, and it changes within centimeters of, like, if you were to break up the soil in depths based on its centimeters, you're going to have different soil chemistry. You're going to have different root morphology, and of course, you're going to have different soil biology. So a way to test that would be literally to get like a soil core, take out your soil core, break it up, and then do like a diversity profile of each different section or depth of soil. Now, that's not going to necessarily tell you the function. That's not going to necessarily change the function, because you won't be able to test it in real time. But what it will do is it'll tell you the types of microbes that are there, and then you can kind of backtrack and go, right, well, we didn't see a lot of, at 400 mil, we didn't see a lot of soil biology. They're not penetrating that deep unless the roots are going that deep. Then the only kind of soil microbes that you're going to capture are ones that exist in bulk soil. So I had a slide before that had like the bulk soil, the rhizosphere, and then so the majority of beneficial soil microbial functions exist around that rhizosphere, rhizoplane, endosphere area. So if the roots aren't going that far, and even if, like, let's just say you got like 4 or 5 different crops going into the one paddock, so over the space of a decade, and you've rotated them correctly, you've done cover cropping, all that sort of stuff. If a root doesn't penetrate further, so whatever the deepest root goes, whatever crop you're using, the soil biology will probably only be affected to that deepest level in the soil profile. There will still be microbes that exist further down. Their function, we're not really sure, and how long they can lay dormant and feed off. Because again, you lose carbon, you lose nitrogen and water through leaching in soil. So further below the root system, you still have these microbes that are feeding off carbon deposits, nitrogen, and water that does actually fall through that hard top. Not a lot, because obviously a hard top being a hard top, there's only so much physical space that things can move through, and the more compacted the soil is, the harder it is for those deposits to move through. And that's why that I was saying before, that soil aggregation is very important by microbes, because they create those pores, those air pockets, water pockets, nutrient pockets, whatever it is, so that they can move freely around the soil. And so farming equipment, so the tractors got the same. If you ever look at an AB line of a field, if those headers and they've been going over the same tracks over and over and over again, directly underneath those tracks, and you can even see it. There's not a lot of growth on those lines, and it's not necessarily none, but there are definite drops in yield along those lines, and that's because those roots can't penetrate deep enough to access the nutrients and the microbes that they need. So in terms of your question about whether you could develop a project around it, I'd have to have a think about it, but it'd be a fascinating one. But again, my first thought would be just to do soil coring, split up your soil core into depths, and then run a diversity profile off each depth to see, all right, obviously at this point you've ripped it up, so they've done the deep ripping, and that you might have increased the bacteria and the fungi short term because you've just exposed them to a whole bunch of carbon that they didn't have before. If you go back to that same spot and take another sample in 2 months time, 3 months time, is it going to be the same? Are they going to have the same function? Are they going to be at the same levels? I personally don't think so, unless you've taken care of that site by adding more carbon in, more fertilizer, whatever, or you've put a crop in that they can then benefit off. And then again, that crop that you've added in, so it's been a fallow and then you've ripped it up and you put your crop in, that crop might benefit from it for a short time. But then, your second, third, fourth year, are you getting that same benefit of amount? Are you, are those crops putting in anything, or are they taking? Most cash crops in Australia take more than they give.
Yeah.
So more often than not, you might do a cover crop over the summer or over the winter, whatever you're trying to grow. If you're doing a summer crop, you might put your legumes, over the winter, and that's great for nitrogen and maybe a bit of carbon. But then your cash crop comes in and takes all that carbon out, and then you're back to a deficit. So you sort of on the merry-go-round there. Yep. So how can we set up a section or a system that lets us have a net positive carbon result? We don't know. There's still a few things at play in terms of the microbial space. We're still unsure at exactly what mechanisms are at play to increase that carbon, and that's why I'm doing what I'm doing. So,
do you think we'll be going back to the pasture lay? That's what we were doing before we had fertilizer. Growing pastures to then build up your soil carbon, so then you could grow your cash crop that would then, deplete that soil carbon. So Southern Cross University has just introduced a regenerative agriculture degree. Now, regenerative agriculture basically is any kind of practice that allows you to have profitability both economically, and in the soil. And to me, I've seen a lot of regen ag work, study, practices for livestock and pastures. I have yet to see any kind of regen ag practices, principles for cropping. And I don't know whether it's, there's obviously going to be ways that we can do it, but it's not going to be a one size fits all principle. It's not going to be able to be a blanket set of system rules for all of Australia. Within, 100 square kilometres, you've got completely different soils, completely different crop management practices, completely different crop types. Here on the Northern Rivers, you'll go from sugarcane and right next to it, you've got a whole bunch of macas. How can you employ the same regen ag principle to those two crops that are so genetically and different? And then the soil biology around those two crops, are they the same? We don't know. We haven't tested enough diversity profiles of different crops to figure out whether those two have enough of a layover, like a crossover in terms of the types of microbes that are doing that function. If we find a heap of the same species that are doing the same thing over and over again, then we can, combine principles to crop species.
we may have to resort to giving our paddocks a bit of a break. And this is why I keep going back to the analogy of having the whole country switch to solar energy overnight. It's not going to happen. It can't happen. There's too much at play. So to get the whole of the country to switch to sort of biological practices and reduce our sort of fertilizer and chemical input, it's not going to be an overnight thing. It's definitely not going to happen within a short period of time. It's going to take an individual grower approach. And that is, so hitting the growers one by one does take time, but it's the most accurate way that we'll be able to, it's not the most efficient way, but it's the most accurate way that those growers are going to get the most out of their fields from their soil biology, because there's going to have to be a way that we can first assess their soil and then design a regime for that grower to say, this is what you should be doing to your soil. They're doing it for, DPI have the agronomy package research team, so like the cereal agronomy team. They're developing agronomy packages for growers to say, this is the wheat type that you're going to grow, this is the sowing window you need to grow it in, this is what you should be doing to your wheat beforehand. I'm saying, why can't we do the same thing with our soil biology, in our microbes, in our soils unit? Why can't we develop, this is your soil type, these are the crops that you're trying to achieve, this is your average daily rain, like yearly rainfall, this is how your temperatures and your soil pHs, then these are the microbes that you should be targeting. And this is how you can help you both naturally target those microbes. And if you can't naturally do it, then here's a few inoculants that will help you along your way. And again, it might not be perfect, but it's a start.
Yeah, great. All right, thanks, Cameron. Well,
No worries.
We've taken up well over the half an hour that I think I booked you in for. Good, nearly hour and quarters. Sorry,
Brendan. As I said, once you get us PhD students talking about our research topic, it's very hard to shut us up. So I think this is where those beers come in handy, mate. You just have to give us a beer to stop us from talking.
Absolutely.
Yeah, no, it's all good. I think, well, I'm sure if Brendan wasn't interested, he wouldn't still be here. So
yeah,
I'm sure. No, it's great that you've hung in, Brendan.
So just another quick one, Cameron. I'm just reading an email here from Mick. He was saying next Tuesday, Wednesday now. Do I need to call Mick about this?
What I'll do is we're about to have a meeting. So I'll have a chat to him, and then we'll get back to you about exact dates and about sampling numbers and all that sort of stuff.
All right. So I just got here. I'll just read it to you. It says, hang on. Sorry. It can be available. So he's dobbed you in. He said, I can be available with Anders and Cameron. That's as in you're doing it. Next Tuesday or Wednesday. Yeah. That's Wednesday next week.
Yeah.
Or the following Tuesday, Wednesday. So no, definitely next week would be the go.
Yeah. For me, I think ideally we'd be coming on the Wednesday afternoon and then going home on the Thursday.
That's what I thought.